Hello, everyone, and thank you for joining us for Webinar Wednesday, even with the slight scheduling change. Hope y'all aren't too disappointed y'all have to listen to me today. We'll give everybody a few more minutes to potentially log in, and just some quick housekeeping while we're waiting for a few more minutes. Don't forget to fill out the post-completion survey. I'll have that link here at the end of the presentation. I'll also throw it into the chat, and you'll see it in your follow-up email that you'll get as well. As much as I would love to claim clairvoyance, I don't always know who iPhone or owner are as far as names that pop up on the chat. So we really do need that completion survey to show that you were here with us today for Webinar Wednesday. And we would hate to not give you credit for Master Dairy. So today we are going to talk about udder health and structures for both large and small ruminants. If y'all have any questions throughout, feel free to throw them into the chat, and my trusty extension specialist Shepard Stearns will be monitoring that and can help relay questions throughout. And if we get to the end and there's still some questions, feel free to unmute and ask them at the end, or use the raise your hand function and stop me while we're going. For those of you who haven't met me yet, I'm Dr. Liz Eckelkamp. I'm the Dairy Extension Specialist here at UTIA. And to save on bandwidth, I'm about to turn off my video, and then you'll just have to listen to me for the next hour. So quite obviously, with udder health and structures, we are going to start out by talking about the udder, and we're going to talk about it in both large and small ruminants. So for most of my life, I've been very used to the traditional cow udder, and I'm getting more and more familiar with the sheep and the goat udder. And there are some major similarities, but some really big differences between the two when it comes to how we manage the small ruminant udder versus the large ruminant udder, and what some of our critical control points need to be between the two. But most of the basics are the same, but there are a few differences. So today we're going to talk about the mammary gland and its structures, because we need to know a little bit about how the mammary gland is put together before we can start trying to protect it, the types of mastitis that we see in large ruminants and small ruminants, and believe it or not, there are more similarities than differences, and some of the keys in prevention that we need to think about for both species, and how that can translate beyond the animal and even out into our parlor and how we manage our parlor, and even something as simple as our wash system. Most of you probably already know these terms, but just in case, mastitis is not actually a disease. Mastitis is really the inflammation of the udder. So anytime we see those inflammation responses, so red, swollen, potentially some changes in milk constituents, that's what we classify as mastitis. We also want to pay close attention to our somatic cell count, or SCC. And again, most of you are probably already familiar with this term, but that is our fancy dairy term for white blood cells that are residing in the udder. So they're still white blood cells. They are not pus. They are white blood cells, and there are normal circulating levels of these, just like there are of red blood cells. But when they're in the udder, they're somatic cells. Milk quality is monitored through somatic cell count, bacteria count, and absence of impurities. And that would be things like flakes or clots in the milk being a different color, a different consistency, and making sure that we don't have any blood in the milk from trauma to that teat or trauma to the udder. The other ones that we probably see a lot on reports, but might not necessarily dissect down to what these acronyms stand for, is SPC, or our stranded plate count. That is the total colony forming units that we can culture from milk. And it's not really bacteria specific. It's just everything that's in there is our standard plate count. Our PI is our preliminary incubation count. And that's looking at holding that initial milk sample a little bit longer on that plate. And I believe that one's held at refrigeration temperatures, but we will get into that a little bit later on. And then LPC is laboratory pasteurization count. And this is an approximation of how many bacteria will actually survive pasteurization and be in there to colonize the milk after we pasteurize. So these are some really important bacterial tests that we do to make sure we have high quality milk going to a consumer or going to our families if we're using these animals for single consumption. We do have some small ruminant specific terms, And we really do not use these as much in the dairy cattle industry, even though we may see these things. And one of those is blue bag. And blue bag is a lack of oxygen reaching the udder itself. And there's actually a bluish tint to that bag. And that could be from disease, lack of, circulation, but we do tend to see these things in relation to mastitis because of some of the changes that can happen with certain types of mastitis and almost being more severe in small ruminants than they are in large ruminants. The other one would be hard bag, and this is physical hard nodules inside the udder from a current or previous mastitis case. We usually think of Staph aureus as a culprit for a hard bag in dairy cattle or small ruminants, and that's honestly that scar tissue that comes from a previous mastitis case. If we think about milk quality, we have to think holistically. There is not just one space that bacteria come from. There's not just one space that pathogens come from. And all pathogens are not going to affect all animals equally, depending on their state, depending on management, and depending on the control points that we have in the parlor. So because of that, we have to think about the animal, how we manage her, what our milking procedures are, and what kind of steps we take to prevent issues in the udder. We have to think about parlor management also, because it isn't just at milking where we have to think about the parlor. We have to think about our cleaning. We have to think about our vacuum rates. We have to think about our flow rates. We have to think about pretty much everything. And equipment cleaning as well. And this is going to be part of the parlor, part of the bulk tank, all of our lines, anything that has potential to touch that udder, we want to keep extremely clean. And then milk cooling as well. Are we getting that milk dropped down to temperature quickly and efficiently enough that we're not giving bacteria time to grow? With the udder, again, terms you probably already know, this is what we refer to the mammary gland as colloquially in the dairy world. So it's not really the mammary gland, it's not really the mammary system, it's the udder. And within this udder, we have 4 quarters in a dairy cow that are fully distinct from each other. Nothing crosses over internally from one quarter to another. There's actually some pretty cool things you can do with dye where you can insert different colored dyes into different quarters, or in the case of small ruminants, different halves into a cadaver udder, and you won't see any crossover. So one quarter will be dyed red, one green, one blue, one yellow, whatever you want your colors to be. And that really helps show us the delineation that what happens in one udder or one quarter is separate and distinct from the other, but there can be transfer maybe through the blood barrier or from teat to teat. So external transfer is where we really start to think about problems, not so much internally from quarter to quarter or from half to half. And this is our moneymaker because this is where our bread and butter, quite literally, is going to come from. The udder is what creates milk. But we're going to see in some future slides that it's not just this big open container where milk just proliferates. There are some very specific things that have to happen in order for us to get milk from that secretory tissue into the udder, out through our canals and down into our teat, and ideally into our milk system, or if we're milking by hand, into our bucket. The other thing we don't always think about with the udder is that it is our best defense against bacteria entering the milk supply. So we really want to make sure that we do everything we can to maximize that natural protection that the udder is able to offer. Just some quick overall anatomy. If we think about our cows or our ewes or our goats, what we're going to see is that that is centrally located towards the back end of the animal. So here's our cow, here's our ewe or our she-goat. And what is similar about these is that there is one canal or cistern. So we have this one gland cistern, this one teat canal per teat. So we don't have kind of this clustered individual system that we're going to see with a sow or a mare. And we don't have 2 canals per teat like what you can see with a camel. So really, per quarter, there's one way in and there's one way out. We also want to think about how that udder is actually attached to the animal. And we usually think about these things from a judging perspective or from a type perspective. But physiologically, these ligaments are extremely important. Now, we don't often see bras on our cows or on our sheep or on our goats. They do have them. I have seen them, but they're not a very common function for our dairy animals. So instead, we rely on that natural ligature to make sure that our dairy animal's udder is firmly attached to her underside and has less chance for damage and less chance for any kind of bacteria to be introduced. So that first one that we want to think about is that lateral suspensory ligament. That's this number 1 over here. So you can see this ligament literally runs down the side of the udder. And what that's going to do is hold those quarters or those halves in together. So that keeps those structures tightened into the center. The one that it's really easy for us to see externally is this number 2, this median suspensory ligament. And we usually refer to that as the MSL because median suspensory ligament is a bit of a mouthful. So if you ever hear someone talking about the MSL, they're talking about that external structure, and we're going to skip a few ahead because I have a good picture of it, I thought. Maybe not, just kidding. All right, never mind. We'll get to that here in a minute. If you look externally on, we'll go back. If we look externally on these goats right here, you can kind of see where that cleft exists on the back end of that udder. And that sharp cleft, that sharp divide between halves, you might even see it better on some of these other pictures. Yeah, the sharp divide between halves is what we're talking about is that median suspensory ligament. And this median suspensory ligament is what actually holds the udder up and into the body cavity of the animal. So without that strong cleft, without that strong median suspensory ligament, that udder is going to continue to sag over time. So if we have a first lactation animal, so a first calving, lambing, kidding animal, first baby, and that median suspensory ligament is not fully evident at that point, that ligament isn't going to tighten up. It's not going to get better over time. So we want to have that strong median suspensory ligament from the get-go. That way we know we have a long lifetime of production from that animal. Because if that median suspensory ligament is weak, we're going to get one of those pendulous dangly udders that we tend to think about in our older animals. And that can lead to potential for increased trauma by catching it on a structure, stepping on it with a hoof, or even having it stepped on by another animal. And we can actually rub these teat ends into the side of their legs as they walk. If they have a poor lateral suspensory ligament and a poor median suspensory ligament, those teats kind of flower and bow out to the outside. And what that can do is increase our bacteria exposure risk and really increase our chance for mastitis. That next thing you want to think about is the teat end. We can see here with that teat end, y'all can see this pretty clear picture of the teat cistern here, or the teat sphincter. And what that's going to do is that teat canal and that teat sphincter are going to keep milk into the udder until we're ready to milk her. And they're also going to keep bacteria out. You can also see from these structures, we might see it just as well. That's better in this picture. Go back. You can see from this structure here, this is the teat cistern and the gland cistern. So we see these big open wells, these big open areas where milk can actually collect. For a dairy cow, there's maybe a cup of milk in each one of these teat canals and gland cisterns at just a normal resting state. All the rest of the milk is actually up here in the secretory tissue. So in order to get it out, we have to maximize oxytocin, that hormone that is responsible for milk letdown. So if we're shouting, if we're screaming, if we're causing any kind of damage before they come into the parlor, all of those things inhibit oxytocin and inhibit milk letdown. And we'll see why on this next slide. So we already talked about the teat sphincter a little bit. That's this number 1 right here. And this is an enlarged picture of it. So this is what will actually close. The teat sphincter is what closes and keeps all the milk in the udder or lets milk pass out of the udder. This is our teat cistern and our gland cistern. And up here in 10 is where we're going to see the rest of that secretory tissue. And that secretory tissue is made up of thousands and thousands, if not millions, of alveoli. This is a super enlarged picture of an alveoli. Each one of these epithelial cells in here is where milk is actually created. So these epithelial cells each create maybe the amount of milk of the tip of a push pen, not a writing pen, a push pen. These myoepithelial cells are stretchy on the outside of these epithelial cells and actually contract when they're exposed to oxytocin. So we're literally squeezing tiny, tiny droplets of milk out of each one of these epithelial cells that's going down into these little ductules, which eventually will become larger ducts that travel to those cisterns, either in the gland or the teat, and allow milk to pass cleanly and quickly out of the udder. And this is why those milking procedures and that letdown time are so important, because if we mess up the oxytocin pathway, there is nothing happening at these myoepithelial cells. So nothing is coming out of these epithelial cells in the alveoli. So all we end up doing is emptying this gland cistern, emptying this teat cistern, and then we have vacuum pressure essentially on something that has nothing left to give us. So the explanation that I'll use a lot with my 4-Hers is, have you ever put a vacuum hose up to your arm? And most of them laugh and giggle and then eventually say yes. Well, and if you leave that vacuum hose on your arm long enough, you're going to get a red spot where that vacuum pressure is trying to pull against a system that has nothing to give. And that's what we'll see happening to our teats over time. That's where that hyperkeratosis comes from. That's where that teat damage comes from, is trying to pull a liquid out of a system that essentially is bare at that point. So that's why that 60 seconds, that 120 seconds from, the first time that cow sees the parlor to the time that we attach the unit is so critical. That's because that's how long it takes a signal from the teat to go to the brain, say, hey, guys, time to release oxytocin. That's how long it takes for oxytocin to actually get back to the udder and start working on these structures. And if we miss that window, it will restart. But for a period of time, we're essentially milking an udder with nothing to give us. So we can cause a lot of damage in a small amount of time if we go outside that body's biological capability. And I did have that picture on here. Good job, me. All right, so we talked about already, the mammary gland is a closed system. The only way in is crossing the blood barrier, and that's how somatic cells get to the udder. They're not naturally there. They're coming in response to an infection. So if we cut our finger and we have, that raised puffy area, we're gonna have more somatic cells or more white blood cells, right, in that area because they're coming in response to a trauma. They're coming in response to bacteria. Same thing with somatic cells. The other thing that we're gonna see is entering through the teat end, and that's usually how pathogens are entering the udder, is coming in through the teat end. Well, not usually. That's kind of the only way in, unless we have a supernumerary teat that manages to actually go all the way into the udder with its own little mini teat cistern, but still through a teat end, just not one of our primary 4, primary 2. We already talked about this inflammation of the mammary gland, and it's caused in response to different stimuli. So the main one that we think about in the dairy industry, whether we're talking about sheep, goats, or cows, is bacteria. And this is a gram stain of Staph aureus. If you've ever had this one in your herd, you remember it because it's a little bit nasty. We don't like this particular mastitis pathogen because once it gets into the udder, it is incredibly difficult to cure a case of Staphylococcus aureus mastitis. It's incredibly difficult to kill that bacteria. And we'll talk about why a little bit later. The other one that we don't always think about is yeast. And this is yeast over here budding out. And that's how we can detect that it is in fact yeast and not bacteria or algae that are causing that case of mastitis. And this one is usually our fault. And we'll talk about that a little bit later. And next is algae. Algae can and will and does colonize the udder, which is why if you've ever been to one of my talks and we're talking about udder health and we're talking about housing, one of the first things I tell you is absolutely no ponds, because algae does not respond to antibiotics. Once it's in there, it's there, and it will continue to cause issues, continue to cause trauma, and continue to decrease the amount of milk that cow is able to produce and reduce your overall farm efficiency. We can have inflammation of the mammary gland in response to trauma, and that would be getting a teat caught in something, having a teat stepped on, or in the case of nursing lambs or kids, or maybe even a nursing calf, getting butted too hard by that offspring, or even getting bitten by that offspring could cause some trauma. Mastitis is far and away the most costly disease in the dairy industry worldwide. It does not matter which country you're in, how many cows you have, what breed you have. Mastitis is going to be the most costly disease you have in your operation. And it's not always due to those visible costs like trying to treat it with antibiotics and having to dump milk for a period of time. Mastitis actually decreases productive capacity of that animal for the entire lactation. And in some cases, with some types of mastitis, it decreases all future production as well. So it's not just that immediate cost of dumping milk down the drain, it's that 2 to 3% decrease in overall productive capacity. We also have veterinary bills, those treatment costs, and then death in some cases can contribute to that $330 to $430 per case. And that number is only going up because this is based on an older study done back in 2017 or 2016. In a 2007 survey, again, slightly older data, 95% of US dairy herds had at least one case of mastitis each year. And the ones that weren't reporting it probably weren't actively looking for mastitis. And it usually impacts 16% of every herd each year. So with the case of mastitis, a lot of you might already know this, but we're going to go over it anyway. We have a bacterial invasion. Some kind of bacteria makes its way through the teat canal, makes its way past the teat sphincter, goes up into the cistern, and then decides to colonize one of those milk secretory sections. And they're going to multiply, they're going to multiply rapidly. So what we'll see is those bacteria start actually destroying epithelial cells. And you'll remember these epithelial cells are where most of our milk production actually happens. We have to pull it out of these epithelial cells down into our cisterns and out through the teat end. So if we destroy that epithelial cell, there's some amount of damage that alveoli can come back from. But occasionally what will happen is we'll see full atrophy of an alveoli or that alveolus, and it will not come back the next lactation. And that's how we destroy future production from this animal as well. We have some, like E. coli, that do a really good job of letting the body know they are there. Somatic cells come in quick response to E. coli because they have that lipopolysaccharide that's basically a giant flashing light that says, come get rid of me, I am here, destroy me now. There are other ones, like Staph aureus, that can actually survive in the white blood cells that we're sending in there to destroy them. They can also wall themselves off in the udder and wait until conditions become more favorable and shed again. So that's why if any of you are on DHIA test, you might see a cyclical pattern of somatic cell. On this one animal, it goes up, 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 down, 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 up, 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 and it just repeats that cycle all year long, because those Staph aureus are walling themselves off when they're getting attacked and then coming out and shedding again when conditions are more favorable. It's kind of like what we'll see, different method, but kind of similar mechanism to hairy heelwort, right? We'll start to see those spirochetes becoming active. We'll see those heelworts kind of materialize, a digital dermatitis. If we go in there to treat it, conditions become less favorable, it'll retract. And then again, when conditions become favorable, we'll see that heelwort pop back up. So some of these are pretty nasty little buggers that we have a hard time getting rid of. Ideally, what we want is less than 250,000 cells per milliliter across all quarters or across all halves in our dairy cattle or our sheep and goats with our somatic cell count, because more than 250,000 is generally what we assume is happening in response to an infection. Our normal circulating levels of white blood cells or somatic cells in our udder is going to be less than 50,000 per quarter, which is why we say less than 250, we're probably okay. Over 250, they're coming in there for a reason. And usually, if we have a somatic cell count greater than 1 million, and we can see that with visual signs like a California mastitis test in both large and small ruminants, where we see that full gel and it really just kind of looks like jello, we're going to say that's associated with infection in small ruminants or large ruminants. But we'll see here in a minute that there are a lot of questions about what infection status really looks like in small ruminants and when we need to try to intervene in small ruminants. The other thing we need to think about with somatic cell count is it's not just an indication of mastitis in our animal, it's also associated with decreased shelf life of our finished products and decreased quality in our finished products. I had an experience in one of the classes that I guest lecture in this past semester, actually, and it's one that I have never had with milk before, and some of you may have. We were trying a milk, and as soon as it hit our tongue, the entire class makes a face like we just licked a block of salt. It was holy salt like Batman. It was quite a visceral reaction. And the milk was salty, and we know that that is kind of a downstream effect of having a high somatic cell count. And what we'll see there is it's physically changing the structure of the milk in the mammary gland, and that's why we see that spike in electroconductivity. Electroconductivity is responding to increased salinity in the milk, and we can actually taste that if we go beyond a certain somatic cell count. So that's part of that quality metric of 750,000 and below for US herds, and below 400,000 if we're thinking about going out internationally. But there are some real tangible differences in shelf life and milk quality that we can see with a high somatic cell count. We talked about some unknowns in small ruminants, and there are some pretty drastic unknowns. We don't really know which somatic cell count is actually linked to infection. We don't really know what our common pathogens are, particularly in the US, and we don't really have great uses for treatment because in the US, there are no labeled intramammary antibiotics for sheep or goats, which means anything we have access to on the market, we are going to have a veterinarian write us a prescription for, and it's going to be off-label use. So for any of my small ruminant producers on, make sure you have a good relationship with your veterinarian, because we're still going to need to treat for mastitis. It's still going to happen in our small ruminants, and we want to make sure that we can effectively treat those cases when they occur. For our cattle, we have some very defined somatic cell count thresholds, and we've been using these with DHIA testing for years, and there is a clear association with milk loss. So as we go from 100 to 200,000, at 200,000, we see 3 pounds of lost milk production capacity in that animal. As we continue moving down the scale, as we get over 1 million, our losses become even more substantial. So this is a loss of 12 pounds of production to 15 to 18. Now, I don't often see things over 640 million, 6.4 million, but we do see over 800,000 in quite a few of our clinical cases of mastitis, particularly with some of our coliforms. Again, because somatic cells can detect them very easily. They do a good job of alerting the body, which is good. We don't begrudge them that, but we want to stay below this somatic cell count as much as possible. So we want them to get in there, do their job, and go back down. Sheep and goats, things are less clear, and production decreases are not really known with our increased somatic cell count the way that they are with cattle. With sheep, we just assume that they're going to be the same as cows, basically is where we get. So if we are below a 4, we're okay. If we're below 250,000, we are not worried about our sheep. If we are above 250,000, we start to be concerned about a potential subclinical or clinical mastitis case. Again, our clinical definition is always going to be the same. We are looking for physical changes in the udder or physical changes in the milk itself. With goats, though, things become even more murky. Below 100,000 is probably healthy in goats, although there is some question about, is it always healthy in goats? Between 100 and 450 is suspect. They might be perfectly fine. They might be experiencing some kind of mastitis issue, but we just really do not know. And a lot of that is because we know there are distinct somatic cell count changes naturally in goats and in sheep along their lactation and across multiple lactations. Above 450,000, we're pretty confident mastitis is going to be an issue, but even then, because we see those differences, we don't really know. So the cutoff we're going to use on some future graphs is 4 for a potential mastitis case for sheep and 6 for a potential mastitis case in goats. And we kind of already talked about this, and I really want to show you this changes over lactation graph. This is a really cool graph that I really enjoyed discovering as I put this presentation together. And down here, we have our sheep. So on our x-axis, we have the stage of lactation in days, And you'll see that it kind of stops at 275, which is a bit shorter than our cattle lactations, but that's okay. And we have our first, third, and fifth lactation for sheep. What we see is they all start off kind of below a 3 at 0 to 50 days, but over the course of their lactation, their somatic cell count naturally increases. And as our lactation number increases, our somatic cell count naturally increases. So if we have a long lactation 5 sheep, she might be over a lactation linear score of 4 just by function of being a lactation 5 sheep. So does she really have mastitis? Probably not. It's just her natural circulating level. If we look at goats, lactation 1 and 2, and even lactation 3, start off lower, just like everything else, which is what we would hope that they're, lambing or kidding in with a lower somatic cell count. But even then, they are naturally higher than both our sheep and our dairy cattle are hopefully ever going to be. And as that lactation progresses, we just get closer and closer and closer to a linear score of 6. So even our first and second lactation animals, at about 250 to 270 days in milk, they are naturally going to be over that somatic cell count threshold. And for our lactation 3 animals, they're going to be at or above that threshold starting at around 125 days in milk, which is about half of their lactation capacity. So we can't really use that linear score as our only metric for detecting when mastitis occurs. We're going to have to be really vigilant with looking for secondary signs and making sure that once it goes clinical, we are ready with a plan to try to fix that issue. Trying to categorize it based on the literature, there's a lot of disagreement, and it's because it depends on what kind of decision that we're trying to make. So if it's an extreme decision, like culling, we're probably going to go with a higher end decision on when we're going to consider this a somatic cell count problem, because that animal has completely left our flock or completely left our herd. However, if we're trying to dry treat that sheep or that goat to try to prevent a future issue, which again is still off-label use, y'all, make sure you talk to your veterinarian, we're probably going to go with a lower level, because just in case anything is there, we want to go ahead and dry treat, provide that intramammary antibiotic, provide that teat sealant to try to knock down any future issues in that next lactation when they lamb or kid back in. If we're trying to identify subclinical mastitis, that's where a lot of the disagreement comes in, because we don't want to give them an off-label drug, because that's what it's going to have to be, if there's not an active case of mastitis. So at that point, trying to find a veterinarian or a lab to work with in your area that can take those milk samples and culture them out is going to be really important, because if we don't see active bacteria present, we don't need to treat with an antibiotic. The antibiotic is only going to do something if there are bacteria there for that antibiotic to kill. And if it's yeast or if it's algae, not much we can do. They're antibacterial, not anti-yeast or anti-algae. We have 2 overall classifications of mastitis, and we will see how much of this we get done. I probably have a little bit too much content in here, but we can always revisit this presentation again in future webinar Wednesdays. Our clinical is visual or clinical signs, and those are flakes, clots, or changes in appearance. So we can see that there is a distinct difference between this left rear quarter and this right rear quarter, right? This one is enlarged, it looks inflamed. It just looks angry. So we're pretty sure there's something happening in that quarter to make it angry and to make us concerned that there's a case of mastitis. Over here with the sheep, what we're seeing is inflammation or maybe even blue bag happening in the sheep, which is a clear clinical sign change from what a normal, healthy udder would look like, as opposed to subclinical, which is where we have no visual signs. Our money maker looks perfect. We're not concerned about anything, but there is an increased somatic cell count. And aside from those natural changes that happen in sheep and goats, the only increase that we should see in somatic cell count, particularly for dairy cattle, is if they're coming in response to some kind of invading pathogen, whether that's bacteria, yeast, algae, or trauma. That's the only reason those somatic cells are going to increase. We have 2 overall groups of mastitis, just like we have 2 overall types of mastitis. Our first one is contagious mastitis, and this is one that is spread from animal to animal. So they don't just exist naturally in nature. They're going to go from one host to another host to another host. And some of them we can control really well, like Strep agalactia. That one we've done a really good job of controlling because it responds well to antibiotic therapy. And some that do not respond well to antibiotic therapy and kind of spread like wildfire, like Staphylococcus aureus. Then we have others that are environmental. They are just endemic to the environment. They just exist in nature, and we all live in nature. So exposure risk has to be limited in other ways besides putting them in a plastic bubble, which does not work all that well. We can find them in our bedding, in our pasture, in the water, in other animals, or even things like pest species like flies can carry some of these environmental and some of the contagious. If we think about contagious mastitis in cattle, our four main culprits are Staphylococcus aureus, which is probably the one that we hear the most about because of that cyclical pathway. And because we've known about it for so long, we know there are some real problems with Staph aureus. We don't talk as much about Streptococcus agalactia today as we did 20 to 30 years ago when this was a major pathogen. Like I said, it responds well to antibiotic therapy. If we ever culture it in a herd, we know what to do with it and we can fix it pretty quickly. Mycoplasma species are another one that's tricky. We can't culture them well on a traditional plate. They need a special kind of media. But we can see some other downstream effects that are a problem with mycoplasma besides just mastitis. And the other one we hear about now more than we used to is non-aureus staphylococcus. Basically, staphylococcus that is not Staph aureus. We used to call these CNS or coagulase negative staphylococci, but we've learned a little bit more about these species, and they don't all coagulate. They're not all negative coagulation. So some Staph chromogenes species will still coagulate, and it gets incorrectly identified as Staph aureus. So because of that, now it is non-aureus staph. We used to also call these opportunistic. They're only going to pop up when everything else is under control. Well, we're learning more about them, and that's not really true either. So if we start to see a lot of NAS in our herd, we need to be concerned about it spreading from animal to animal, just like we're concerned with Staph aureus, just like we're concerned with Strep ag. And these can spread through a couple of different ways, but one of the main culprits is milking time, because a little bit of milk will be left on the milking machine inflations or on the hands of milking personnel, which is why we always stress, I don't care how many animals you are milking, it could be 3, it could be 3,000, your milkers need to wear gloves. Our hands are a great place for bacteria to grow. They do a really, really, really good job of keeping things like Staph aureus alive and kicking, and these NAS species alive and kicking. They love our hands. It's kind of annoying. They love gloves less. So if we have a really, really dirty set of gloves, is there still potential to pass it off to another animal? Yes. Is it diminished? Absolutely. The other one that we'll see is dirty udder towels or using one towel for more than one animal. So I can't stress this enough. Gloves on all of our milkers and one towel per animal. I even get kind of squirrely about using a single towel to do brush off of debris on multiple animals, just because if we get a little bit of milk on that, and that milk has Staph aureus, we've just now introduced that to every single other animal down the line. I'd almost rather you brush them off with your glove, wipe your glove off afterwards, and then go back and start your regular milking routine. The other one that we can't control as directly is flies. If flies go light on an udder of one animal, they can actually bring back some of those contagious pathogens like Staph aureus or non-aureus staph and bring them back to another udder of another animal. One that we don't see as much in large ruminants, but we do see in small ruminants, is being spread from nursing offspring back to the dam. And one of our main culprits is the same, Staphylococcus aureus, and this one in small ruminants is nasty. Like, we don't really like it in our dairy cattle, but it doesn't usually make an udder half slough off. So that picture that we saw at the very beginning of the slide deck where there was that big hole in the udder, that's caused by Staph aureus mastitis in small ruminants. So small ruminant people on here, pay really close attention to Staph aureus mastitis. It can cause some major damage. And one of the recommended ways to try to treat Staph aureus mastitis is literally to take off that entire udder. Amputating the udder is a legitimate way to try to control Staph aureus mastitis. And if we're amputating that udder, we can't get any milk from it ever again. So we want to pay really good attention to prevention on this one. The other one is Pasteurella hemolytica. We don't really see this in our large ruminants, but we do see this a lot in our small ruminants. And with our Pasteurella species, what we'll see is that's actually spread from lambs or kids that are experiencing a bout of pneumonia, and they will then inoculate that teat end when they nurse on mom. We also have environmentals, and these are pretty much the same regardless. For our cattle, we have our gram-negative species. The two we probably think about most are coliforms, which would be E. coli and Klebsiella pneumoniae. Those are the two that we really tend to think about as far as a problem in cattle. From a milk quality standpoint, that EHEC 0157, which is, I'm not going to say it because I'm going to mess it up, but basically that's the version of E. coli that can cause kidney failure. And pasteurization denatures that EHEC 0157. But if they drink milk that hasn't been pasteurized, that's the one that causes some major damage. That's probably one of our worst foodborne pathogens. We also have our environmental streptococci, and those are Streptococcus uberis and Streptococcus dysgalactia. These guys just live in nature. They do respond well to antibiotic therapy. Gram-negatives probably don't really usually need antibiotic therapy because the body is really good at finding them on its own. We also have yeast, molds, and algae like Protheca that comes from ponds, which I hate, so no ponds for your animals. We also have bacillus species that can come from the environment, particularly if our bedding is really wet or if we have a lot of teat end damage and we're bedding on some kind of wood byproduct or straw. Having that damaged teat end makes it much, much easier for that bacillus to infiltrate that udder. And bacillus is really cool for me to watch grow on a plate because it really just grows out like this weird scabby spiderweb looking kind of stuff on a plate, but you really don't want it in your herd. It will kind of respond to antibiotic therapy, but still not something that we want to look for because it's an off-the-wall pathogen. So if you're sending in samples and one or two of them come back with bacillus, we'd probably usually call it a contaminant, but it can actually cause cases of mastitis. With our small ruminants, we're not thinking quite so much about those bacillus, and we don't have as many documented cases of those off-the-wall ones like yeast and algae, but we do see quite a few of those gram-negative species, those same culprits, that E. coli and that Klebsiella are culprits for everybody. Same thing with those environmental streptococci, that's Strep uberis, that's Strep dysgalactia, culprits for everyone. And this does not mean that your small ruminants cannot get algae as a cause of mastitis. They absolutely can. So no ponds and no really wet, nasty, muddy areas either. Yeasts are generally acquired during mastitis treatment, which seems a little counterintuitive, but we can introduce yeast when we're giving that intramammary antibiotic therapy and we don't fully clean the teat end. So that means dipping it with an effective pre-dip, wiping off that pre-dip, and then using individual alcohol pads to fully scrub that teat end clean until we ever think about introducing the tip of that intramammary therapy to that teat end. We also don't want to use the same tube on multiple teat ends. One tube per teat, whether she's got 4, whether she's got 2, 1 tube per teat, because we don't want to cause a problem by using that same tube that could end up costing us a lot more money in the long run than that extra tube of intramammary antibiotic or that extra tube of teat sealant. 1 tube per teat. These non-ORA staph, I didn't change this one, they are not really opportunistic. We already talked about that, need to update that slide. But for small ruminants and also in large animals, what we're seeing is these cause the majority of mastitis infections today. Most of our mastitis cases are actually coming from these non-ORA staph. Not so much our staph aureus, not so much our environmentals, really these non-ORA staphs. We've already talked about decreased milk production, decreased product quality, decreased shelf life. One thing we might not always think about from a small ruminant perspective, probably more so than a dairy cattle perspective, is on that lamb or on that kid, or if we are nursing an animal on our dairy cow, decreased average daily gain on that offspring. If we have mastitis cases, we're producing less milk overall. We already talked about that, but we can produce so little that it starts to affect the growth of that offspring. So even if you have meat animals, even if you have beef cows, even if you have meat sheep or goats, mastitis is still a really important disease, and we want to try to protect that udder as much as possible. We really want to have layers of protection. So we want to manage their environment. We want to clean and disinfect the udder and clean and disinfect the milking equipment. And we're just going to pass through this really, really fast. We already talked about those. And really the keys to contagious are trying to separate those animals that are infected. So strep ag does respond to antibiotics. Staph aureus really doesn't. So whether we are in a dairy cattle herd, whether we are in a dairy sheep and goat herd, culling is our best solution. I believe Dr. Strickland calls it trailer mycin. Get them on a trailer, get them off the farm, because if they're still there, they're going to continue to shed and continue to expose other animals in your herd or in your flock to Staph aureus. And once it spreads, it's really hard to get rid of. So losing that one animal to save the next 10 really can become your most economic decision. Isolating infected dams or offspring. If you are bound and determined to keep them in your herd, milk them last. Know who they are, know what group they're in, try to prevent mixing them with other animals, and they're the last ones into the parlor and they go in right before the wash cycle. So our potential to infect other animals is limited. With our environment, we already talked about it. We can't live in a plastic bubble, no matter how much we might want to put them there. But what we can do is make sure that their bedding is clean, dry, and comfortable. And really their barn overall, we want it to be clean, dry, and comfortable. And comfortable is really where that heat stress aspect comes into play, particularly for our dairy cattle, less so for our sheep and goats. That clean and dry area is going to do several things. It's going to reduce bacterial load at the teat end. It's going to reduce material actually attaching to that teat end and reduce additional stress on lactating animals and reduce diseases in adults and juveniles. So if we have nursing lambs and kids, having that clean and dry area is going to reduce our cases of pneumonia, reduce our Pasteurella hemolytica mastitis. If we have lactating animals that have no other choice but to lay in a dirty or wet area, they are going to get stressed. Just like none of us would choose to sleep on a bed that has poop or pee or is wet on it, they would not choose that either. They will preferentially go to a place that is cleaner and drier. And if we have trouble getting in there and cleaning that full area every once in a while, what we'll see is that they'll all kind of turn into a mud pit over time, because they will preferentially move to the area that is not wet, that is not dirty, and then just make that other area wet and dirty. And you really want to common sense check this with a hygiene score, because even if we kind of do a look and say, it's not that dirty today compared to what it was yesterday or the day before, I use that justification in my house all of the time, because I don't want to break out the vacuum, because it takes time and it's tedious, and I could ignore all the fur balls on my stairs, or gathering at the foot of my bed, or just building little colonies in my hallway. And if I see it every single day, it's easier and easier to justify not pulling out the vacuum. But when I look at my socks and they're fully covered in fur, that's like my own personal hygiene score for the house. It's really time to break out the vacuum. And the hygiene scoring card is pretty simple. I'm not mad at a 2. A 2 means that they are out in nature living life. 1 tends to usually be show cow, maybe a little bit of old dirt. What we're worried about is these 3s and these 4s. So with a 3, we're seeing new material that's adhered, but we're also seeing evidence that they've been in a dirty area for a while. And that dirty area for a while is where we really start to worry, because that means that that bacterial load, that exposure, is a whole lot higher. Down here at a 4, what we're seeing is full coverage of things like the teat end, full coverage of the udder, or even full coverage of those rear legs. And those rear legs, when they lay down, are going to go right straight to the udder. If we're on pasture, I know it can be hard to control Mother Nature. She doesn't usually listen. But if we have the ability to, we want to avoid wet and muddy areas, which might mean rotational grazing, rotational pasture access, or when we see one starting to get a little bit too far to the wet side, we can move them to a new area. Or even having a shelter that we don't intend for them to be in all of the time, but having a barn that we can put them up in where we can control that environmental exposure when we can't have them out on pasture. And y'all, I cannot say this enough, absolutely no pond access. They go in the pond, they poop in the pond, they pee in the pond, then they will drink out of it, which boggles my mind, but they will also completely submerge that udder in the pond. If that udder is submerged in water, it does not matter how tight that teat and sphincter is, you are going to get bacteria going into that udder, whether it has 4 quarters or 2 halves. And that Pseudomonas, that Proteus, that yeast, that mold, that Prototheca, that bacillus, they do not respond well to antibiotic therapy. So it's going to cost you a whole lot more in the long run than trying to get water out to those animals a different way or trying to use a different cooling strategy will. Lots and lots of clean bedding and routine bedding maintenance is key. We always talk about sand being king, and that really is just because it's inorganic, which just means no carbon. Not that the one's USDA organic and one's not, just no carbon in the sand bedding naturally. If we use sand bedding and if we don't replace it over time, it is no longer inorganic because the cows have added their own organic matter. So we do need to make sure that we're routinely doing bedding maintenance. We want to pay attention to any kind of special needs area. So whether that's our calving facility or our drop pen or our lambing jug, we want to make sure that that is clean and dry and comfortable, even more so than any of our other areas. Because at that point, both our dams and our offspring are going to be the most susceptible to new issues. And I am vastly running out of time. So the only other thing I want to try to hit on really quickly, talked about wearing gloves, limiting water usage can be really important. Some of the old school ways of milking used to be to fully wash the udder down. Well, if we do that, where does water collect? The lowest point on the udder, which happens to be the teat end, where we don't want any kind of large exposure area. So our new plan is if we use water, I'm not going to yell at you, but whatever water we put on, we have to take off. So that could mean lots and lots of towels per individual animal. We want to make sure we keep a routine that maximizes that letdown, maximizes that oxytocin. That pre and post dip are extremely important, and there are a lot of kinds out there. We have iodine, chlorohex, hydrogen peroxide. Use whatever works best for you on your operation. And if you're starting to see some issues with mastitis cases, look at how effectively you're actually covering that teat end. Are you fully covering that teat end, that entryway into the udder? And are you using one that can stand up to your conditions? If you have a lot of organic matter that tends to be on those teat ends, hydrogen peroxide looks really good. It foams up fantastically, but it's reacting to that organic matter. It's not reacting to killing the bacteria. We also want to limit over milking because of that teat end damage that we talked about before from that vacuum pressure. For our equipment, we want to do a warm water rinse, a detergent wash, and a disinfectant cycle. And even if you're milking by hand and you have some kind of milking equipment that's standalone, warm water rinse, detergent wash, disinfectant cycle. And we're doing that because we want to get any of that old milk fully off of there. We don't want it to build up. We don't want it to proliferate bacteria because that's exactly what we're going to put back on those teats the next time we milk. And that can cause a lot of issues. And we're going to see really quick that SPC and that LPC, the PI, the SPC, and the coliforms. And I don't mind sending these out if you want them. Depending on where our levels are, that tells us where to look. Are we looking at the cows or the equipment or the cooling? We want that milk down below 40 degrees Fahrenheit as soon as humanly possible because it limits bacterial growth. So now that I've gone a really, really long time and very fast over a few things, don't forget to fill out this eval, and that's tiny.utk.edu/WW2022 to make sure you can get Master Dairy credit. Are there any questions in the chat or from the group about all of that information I just dumped on you very, very quickly?
None in the chat.
Okay. Any questions from attendees? All right, well, I guess I blew everybody's minds or I answered all of your questions. One of those two things is probably true. So I'll be sending out the recording later on today. And if y'all have any questions about this that you didn't want to ask during this session, feel free to email me, feel free to call me, and we'll talk about it. Thanks, everyone. Thank you for being here.